And so we'll do that first. Okay, you should see the first slide on coronavirus. Hopefully you do.
Yep.
Yes.
Remind you that I'm encouraging participation, and tell me your name so I'll make sure I record it. So we're in the midst of a pandemic, and I want to give you the basics about the virus and how it causes infection. So here on the left in this slide, you see a scanning electron micrograph, which is famous worldwide. You've probably seen this many times in the news media. And what it shows is that the virus is a spherical particle with these projections on the surface. This is an artificial color here. The projections are red.
Professor, I'm not.
That is the spike protein.
I'm not seeing any picture.
Oh, really?
That's probably on your end. I'm able to see the slide.
I can see it too.
Okay, good.
Anybody but Scott, besides Scott, that can't see it? I guess, Scott, everybody else can, so I guess you have to maybe reboot. Anyway, so inside that spherical particle is, in the center here, you see the red squiggle is the genome of the virus, which in this case is RNA. The particle itself is encapsulated in a membrane, so there are a number of proteins and other molecules involved there. But the critical thing is that this spike protein protrudes out away from the virus particle itself. And that's critical because that's the part that attacks human cells. So what it does is attach to a receptor on the surface of human cells called ACE2. ACE stands for angiotensin converting enzyme 2. And I'll tell you what that does in a minute. So this is a phospholipid bilayer. ACE2 sticks out from that membrane, and that's where the coronavirus binds. Now here on the right, that's a three-dimensional structure of the spike protein, and I'll come back again to that a little later. But you'll see some familiar forms here. You can see that alpha helices in yellow. You can see beta-pleated sheets in blue, and other types of formations. And remember, this tertiary structure is all held together by 4 different types of bonds. So now, when spike protein binds to ACE2, it's taken into the cell by a process known as endocytosis. And that's shown here. So imagine the stars as the virus and the receptor on the surface of the cell. When the binding occurs, a coated pit forms and invaginates and pinches off so that a vesicle is made with the virus inside. And that vesicle can then break and release the virus inside the cell. So this is a normal process. It's a mechanism used by the cell to take up a lot of different kinds of large molecules and cell parts or even other cells. And the virus hijacks that mechanism to get in. So the steps are first attachment, membrane invagination, membrane closure, and finally a virus-containing vesicle. Okay, so what about ACE2? Why is that a receptor for this virus? Well, this slide shows you what the normal function of ACE2 is. So angiotensin is involved in blood pressure regulation. It is converted to either angiotensin 1, which there's another ACE enzyme converts it to angiotensin 2, and these constrict blood vessels, vasoconstrictors, and they cause high blood pressure. ACE2 is a good guy. ACE2 can convert either of these intermediates into a different form of angiotensin, either 1,9 or 1,7, and these have the opposite effect. They dilate blood vessels and reduce blood pressure. So that's what ACE2's normal function is, to lower blood pressure. So what I want to say about this slide is in the center here, within the red circle, ACE2 is involved in blood pressure. SARS-CoV-2, the name of the virus, it's one of thousands of different SARS-type viruses. It happens to have a surface protein called spike, which by sheer chance, sheer coincidence, just happens to have an amino acid sequence that binds to ACE2. So coronavirus does not have evil intent. It doesn't mean to harm people, but it does by chance, just because if you see here on the right, we have the three-dimensional structure of the spike protein, and at the top, the ACE2 protein. If spike binds to ACE2, it starts the process of cell invagination. It gets into the cell, and once it does, it replicates and causes disease. And we'll talk later about why, some of the reasons why SARS-CoV-2 causes such serious disease. But I want to emphasize the point here that this disease, and probably most diseases, are chance occurrences. There are lots and lots of different potential diseases out there, but only a few of them culminate in symptoms. Why is SARS-CoV-2 so highly infectious? Well, there are a number of reasons. The binding of ACE2 to spike is about 10-fold greater than with other SARS-type viruses. And what that means is that far fewer particles are needed to start an infection. So if you are exposed to most SARS-type viruses, you need a big dose in order to become infected. With SARS-CoV-2, you need a 10-fold lower dose in order to be infected. So only a few particles are needed. Now, there is a variant that you may have read about in the news media just recently. It was first found in England, and it has increased transmissibility versus the wild type, which is now called the Wuhan strain, first identified in China. Now, this variant, B117, has 17 mutations in it with respect to the original strain. One of these mutations, N5 501Y. This is an asparagine is mutated, so now the residue is tyrosine. And it binds spike more tightly than the wild type strain. And apparently this tighter binding in this mutant causes it to be more transmissible. Symptoms it causes aren't necessarily more severe, but it's easier to move from person to person. On the second point, there's a host enzyme, a human enzyme called furin that's needed to activate the spike protein. And spike is more readily activated by furin than most SARS viruses. And the third point is that there's a 2-week latent period before symptoms occur. So people can become infected and have a heavy virus load, but not have any symptoms. But while they have this heavy load, they're spreading it around to people that don't realize they're infected. So these symptomless carriers are a big problem. There's one other point I want to make here at the bottom in red, copying errors. So you remember when we talked about DNA replication? It's a complex process, mistakes occur, and mutations occur as a result of those mistakes. And these mutations occur at a constant rate. So every time a nucleic acid molecule is replicated, it's incurring mutations at some rate. So the more the virus replicates, the more mutations there are, and the greater the chance that a new, more virulent mutant will arise. And that's why the CDC, the Centers for Disease Control, are so concerned about the virus spreading and people not taking the proper precautions, because the more the virus spreads around, the more it replicates, and the more chances there are for a mutation that might be uncontrollable by the currently available vaccines.
Yeah, teacher, I have a question regarding this point.
Uh-huh.
It's also possible that currently they are taking some people, they are taking the viruses. It may be the chance of any mutation before that, and maybe it was not effective.
The vaccine is not
The
effective?
vaccine, yeah, because of mutation.
Yeah, that's the big concern. I mean, we've already got a lot of mutations. B117 is just one of them. There's another one from South Africa, another one from Brazil, all of which seem to have different transmissibilities that are very dangerous. But so far as I'm aware, none of them can escape the currently available vaccines.
Okay,
But
that's
the pharmaceutical companies are very much aware of this, and they're continuing to develop vaccines with the anticipation that sooner or later we're going to get mutants that the current vaccines won't control.
Professor, also got a question.
Lathan, Edmund, okay.
When somebody gets COVID, they get an immunity, right? They get like, I don't know, I saw it's like 1 to 3 months immunity. And if they get it like in that time period, do you say they're getting like a different type of the COVID, like a different mutation from COVID, or the same strand?
You mean if they get reinfected?
Yes.
I really don't know
Hmm.
if reinfections are caused by the same strain. Certainly it could be caused by a different strain,
Mhm.
but I don't know if the same strain could reinfect. Scott, do you know? Or does anyone know?
I don't know.
Yeah, from what I remember, and I could be wrong, I seem to remember there being talk about reinfection before we found out about the English variant. So maybe?
Yeah, I don't recall. So I can't really shed much light on the issue.
That's okay. Thank you.
Okay, anything else about why SARS-CoV-2 is so infectious? And
Yeah, I have a question, teacher, about the HIV
what's your name?
virus. HIV virus.
Tell me
My
your
name
name,
is
please.
Mastura Kayumi.
Okay.
Regarding to this changes and mutations, how about the HIV viruses also occurs a lot of mutation constantly, or the about the replication of this, or
Are you asking why do mutations occur?
no, about the HIV virus. Also, these changes occurs in HIV virus because of this. It doesn't have any vaccine.
Right. HIV, even though people have been trying for 10 years or more to make a vaccine, there's no vaccine against HIV. And that's just an example of how it is so difficult to develop a vaccine against viruses. And it's miraculous that the pharmaceutical companies have been able to get one against coronavirus in such a short time, in less than a year. Yeah, HIV is treated by protease inhibitors, not by vaccine.
Yeah, probably a better example would have been like common cold, because that one, it's the reason that we can't necessarily nail it down is because there's just so many mutations and it mutates so quickly.
Yeah, yeah, different organisms mutate at different rates, and certainly the rhinoviruses are among the fastest. But within any organism, there is this constant rate of copying errors. It's unavoidable. And all coronavirus is trying to do is what any organism tries to do, and that is make more of itself. So, and the more of itself it makes, the more variants there are going to be. Okay. Let's see. Okay, so the last thing I want to do with respect to coronavirus is talk about the currently available vaccines and how they work. So there are 3 types of vaccine. I'm not going to talk about the third one because it's not relevant to coronavirus, but 1 and 2 are. So the first is an mRNA encoding the spike protein is translated to spike in the cytoplasm of the cell, and that induces anti-spike antibodies. There are 2 vaccines of this type, one from Pfizer and one from Moderna. The one that I got was the Moderna. They're both very fragile, require ultra cold storage, minus 80 degrees centigrade storage, and they both require 2 doses in order to achieve full immunity. The second type is in a virus vector, and the virus of choice is called adenovirus. It's a DNA virus, and so the spike RNA has to be reverse transcribed to DNA, spliced into the adenovirus genome. It's a disabled form of adenovirus, so it can't cause disease. And then when the host cell replicates the virus, it translates to get spike, and spike induces antibodies. There's one company in the US, there's another one in England. Johnson & Johnson uses an adenovirus vector. Now, this has the advantage that it only needs to be stored at refrigerator temperature, 4 degrees or so. And the other big advantage is that it only requires 1 dose. And so I want to give you just a brief overview of how each of these types of vaccine works. In the case of mRNA, the mRNA that encodes only the spike protein is packaged into a little oil droplet, a lipid nanoparticle. That's what gets injected into your muscle. These little particles fuse to the cell membrane, and once inside the cell, the mRNA is translated to spike protein in the cytoplasm, and the spike gets exported to the outside of the cell where it induces anti-spike antibodies. And then the antibodies block infection. So these illustrations at the edge illustrate this. So in the upper left is a virus particle with the RNA genome. The spike protein is this little red segment. And so it's just this little red segment, just the part that encodes spike, gets packaged into a little lipid particle. And that fuses with the cell membrane and releases the mRNA into the cytoplasm. And this is very familiar to you, the ribosome, the mRNA, the tRNAs, you make the spike protein immediately. This spike protein gets on the outside of the cell, and if you go to the center diagram, where the B cells and the T cells, they're the ones that make antibodies. See the spike protein on the vaccinated cell, and they make these antibodies. These at the bottom of the slide, those symbols are antibodies. And what they do when a virus particle enters the cell, or even before it enters the cell, the antibodies attach to the spike proteins on the surface of the virus and block the attachment. So I can get rid of this. So this is back to the original picture I showed you of spike binding to the receptor ACE2. If there's antibodies attached to the spike protein, this attachment is blocked. And so there's no infection. Is that clear to everybody how mRNA vaccine elicits antibodies and how they prevent infection? Okay. Now I'm going to contrast that to how a viral vector is used to achieve the same result. So we start with, again, the virus particle, and the RNA for the spike protein is reverse transcribed. You remember how that enzyme works? Reverse transcribed to DNA, and then it's packaged in an adenovirus particle. So this is very different from how mRNA works. So now the viral DNA, the coronavirus DNA, is in the form of DNA. RNA has been converted to a double-stranded DNA. And that's what, when it's injected into your arm, that's what your cell takes up, this adenovirus particle. with DNA as the genome of the coronavirus. Well, the adenovirus injects that DNA into the nucleus of the cell. I want to go back and point out that in the case of mRNA, the nucleus is not involved at all. Everything, all the action is in the cytoplasm. Whereas in the case of adenovirus vector, the DNA has to go into the nucleus. And there it's transcribed to mRNA. Now you're back to the spike protein RNA. That RNA migrates into the cytoplasm. And from there, it's just like with an mRNA vaccine, where the spike is on the surface of the cell and everything after that is the same. Once it's on the surface of the cell, it induces antibodies from the B cells and the T cells. Antibodies then will block any new virus that comes in. So do you see the difference between the two types of vaccine? One involves a viral vector, the other is just straight messenger RNA.
I have a question about the viral vector vaccine.
Uh-huh.
This is Dylan, by the way. How do they reverse the RNA to DNA? How does that process work?
Did we talk about reverse transcriptase earlier in the course?
I don't believe so.
Okay, that's my fault. We missed that. I'm glad you asked this question because this is a very important point. The central dogma of molecular biology is that DNA is always transcribed to RNA, and that's always translated into protein. And it's a one-way street. You can't go backward. Well, there was a Nobel Prize awarded to two guys who showed that's not true. That RNA can be reverse transcribed back to DNA. And that ability to reverse transcribe RNA to DNA is very important in biology today. It's used all the time in many different types of labs. So that's what they do to make an adenovirus vaccine. Since coronavirus is an RNA and you want to deliver that in a DNA virus, you have to make it into DNA. I'll try to remember to give you some details about how reverse transcriptase works, since I seem to have missed that earlier on. Okay, anything else?
Professor,
Yeah,
I have a question. This is Kit.
okay.
Why are symptoms different with people who took vaccine?
Speak a little louder, I can't hear you.
Oh, okay. Why are symptoms different with people who took the vaccine?
Guess I don't understand the question.
I think she's asking why are there different symptoms for the different vaccines?
Different symptoms for the different vaccines.
So like the Moderna, you got really sick, but for the Johnson & Johnson, not so
that,
much.
Yeah, that's variation. Well, a lot of that is due to just variation among people. But I must say that the difference between Pfizer and Moderna has what's called an adjuvant, which they mix they put this adjuvant along with the mRNA into the little lipid particle. This adjuvant is there to prime the immune system, to get the immune system poised to make antibodies. Most people who react badly, like I did, are not reacting to the virus, they're reacting to the adjuvant. Pfizer doesn't have an adjuvant in its vaccine. So if you get the Pfizer vaccine, you're less likely to have a bad reaction than if you get Moderna. Now, having said that, not all people who get Moderna have a bad reaction. I have a friend who got Moderna and had no reaction at all, except the injection point was sore. So the main component here in variation is just the differences between people, how different people react to different components of the vaccine.
Thank you.
Okay, anything else? All right. Okay, I want to switch topics completely now and talk about cystic fibrosis for the rest of the period. Before I start this, you may have noticed, or may not, that I put a number of documents in Canvas that are all about the cystic fibrosis project. They're all in the week 6 module, and they're all sort of pre-documents. So I'm going to replace them all after class because I've modified them considerably since I first put them there. The other thing you may have noticed is that I issued an assignment on cystic fibrosis, which is due Monday, March 15th. And the assignment is really like a study guide.
Excuse me, teacher. What about the quiz 5? It includes how many chapters?
The last 2 lectures. So quiz 5 covers DNA, RNA, transcription, translation, protein synthesis, and mutations, and CRISPR.
Thank you, teacher.
Okay, now you see cystic fibrosis introduction, I hope.
Yes.
Okay. The 2 most prominent organisms affected by cystic fibrosis are the lungs and the pancreas. And for purposes of our work here, we're going to focus on the pancreas, not the lungs. But in both cases, the lungs here on the left, there are air passages within the lungs, which get clogged up with mucus for people who have cystic fibrosis. In the case of the pancreas, on the right here, you see the pancreas is in yellow. There's a duct that delivers digestive juices, and the gallbladder here in green produces bile, which is introduced through a duct into the duodenum, which is the first part of the small intestine. these ducts in cystic fibrosis get clogged. So the digestive juices cannot be transferred into the intestine. And in really severe cases, the patient literally starves. And just to remind you, the gene responsible for all this, the CTFR gene, is on chromosome 7. It's on the long arm in region 3, band 1, subband 0.2. That's what the code stands for. Now, the second point, I have 3 sort of unrelated points here to start out. The first point is that of all of the organs that we could be looking at, we're going to focus on the pancreas. Second point is that I want to give you a sense of how a mutation in the CFTR gene causes mucus accumulation in affected organs. So that's shown here, comparing a healthy situation with a cystic fibrosis situation. So in a healthy organ, like a lung or a pancreas, there's mucus on the outside, and that mucus is hydrated. It's thin because of the CTFR protein, which transports chloride ions. Here you see CFTR protein is in green. When chloride ions are transported, so is water. Sodium ions go the opposite direction. And so the mucus is highly liquid, and these cilia on the surface of the cells keep it flowing. And if there are bacteria there, they keep being swept away. In the case of cystic fibrosis on the right, there's no CFTR protein, or if there is one, it's not functioning properly. And so chloride ions are not being transferred. Water is not flowing out. And the mucus becomes dehydrated, thick and sticky. And the cilia, you can see, are having a hard time. The flow of mucus is slow or not existent. And if there are bacteria there, they will, instead of being swept away, they form a biofilm. So this is the problem. It's sort of like a mechanical problem that a mutation in this gene causes, that critical parts of critical organs get clogged up and they can't function properly. And so I have some, yeah.
This is Laisha Sortisans. How does one develop the what's it called? The cystic fibrosis? How does one get that?
Yeah, you've actually brought me to the next point. And that is number 3 here. So your question is, how does one get the disease? Is that right?
Yes.
Yeah, and that's a critical question, because cystic fibrosis is entirely a genetic disease. I've stated it here. Cystic fibrosis is a genetic disease. It is not contagious. It occurs only if both the father and the mother contribute a chromosome 7 carrying the CFTR gene mutation. So that's illustrated in this diagram, where the father has 2 copies of the gene. 1 is mutant. The mother has 2 copies. 1 is mutant. Neither of them has symptoms, because you have to have 2 copies in order to have symptoms. Then in the children, they can either end up with no copies, or 1 copy, or 2 copies. So 25%, there's a 25% chance of mother and father heterozygotes having a child with the disease. 25% chance. So in other words, if they had 100 kids, 25 of them would have cystic fibrosis.
So if the mother and father, they don't have it, the child could still be affected by it? Or
If neither the mother or the father has a copy, none of the children would have symptoms, would have the disease. Child has to have both copies in order to have cystic fibrosis.
Professor,
Uh-huh.
Kit, is there a way to avoid cystic fibrosis disease before it is inherited?
Well, not really. The only way you could avoid it is if you had genetic testing done. If you and your partner had genetic testing and you find that you both have a mutation, then there's a 25% chance that your children, any one of your children would have cystic fibrosis. If, on the other hand, you do genetic testing and find out that one of you does not have a mutation at all, then none of your children would have cystic fibrosis.
Okay, thank you.
Okay, so those are my 3 disparate, sort of unrelated points I wanted to make to start. And now I'd like to focus on the pancreas and the gallbladder. The whole point of this exercise is it's an example, it's to illustrate how a complex process like digestion can be shut down by this disease. So first, let's talk about the pancreas and the gallbladder. So this yellow section illustrates the pancreas. It has this major duct running down to the middle of it. This little bag is the gallbladder, through which produces bile, and the bile enters another duct called the common bile duct. And both of these ducts enter into the duodenum, which is the start of the small intestine. So partially digested food accumulates there. Now, we have 2 terms, exocrine and endocrine. The difference is that exocrine glands release substances out through a duct, whereas endocrine glands release hormones into the bloodstream. And the pancreas has both of those functions, interestingly. And here's how they work. If you take a small section of the pancreas and blow it up, you'll see 2 different kinds of cells. These pink ones with the blue dots are exocrine cells, so-called acinar cells, and they secrete digestive enzymes. That's one type. The blue cells are called islets of Langerhans, pancreatic islet cells, and they secrete hormones. So the hormones are secreted directly into the bloodstream. The enzymes are secreted into the pancreatic duct. So this is what you need to remember. These 2 kinds of cells, one secretes digestive hormone or digestive enzymes, the other secretes hormones that regulate digestion. So the acinar cells secrete pancreatic juice. These are the types of enzymes that these cells produce and secrete. Proteases to digest proteins, lipases to digest fats, carbohydrates for digesting complex sugars, and nucleases for digesting nucleic acids. These are the main components of the food you eat. So the islets of Langerhans are the ones, the endocrine functions, and they produce hormones. You all know about insulin produced by the beta cells. And these are some numbers that you will need to fill out your worksheets. But insulin functions to promote absorption of glucose into cells, and that reduces the blood glucose level. Glucagon does the opposite. It raises blood glucose level by releasing glucose stored in glycogen. It's another hormone, somatostatin, produced by delta cells. It inhibits insulin and glucagon, these two opposite effectors, and that slows down digestion and prevents acid accumulation. And finally, there's the pancreatic polypeptide, which is a regulator of both exocrine and endocrine functions. And again, this is information that you'll need for your worksheet. The pancreas produces about a quart of pancreatic juice each day. And again, the main components are the enzymes that digest polymers in the food you eat. Juice is mostly water, contains salts and sodium to maintain the pH. Now I've got zymogens here in red because it's important you know what zymogens are and why they're necessary. Zymogens are the inactive forms of enzymes. The pancreas produces proteases as zymogens. Now, why does it do that? If the proteases were produced in an active form, they would digest the pancreas itself. So they're produced as zymogens in an inactive form, and they don't become active until they get into the duodenum, where they're activated by enzymes to convert them to active proteases. Is that clear? Everybody understand? The zymogen is inactive and it has to be activated in order to do its job. And the reason it's inactive in this case is because it would attack its own self, so its own organ. The other enzymes produced by the pancreas are not produced as zymogens because they won't digest the pancreas. So here are some examples of zymogens which are inactive. Trypsinogen is converted to trypsin, which is a main protease. Pro-carboxypeptidase to carboxypeptidase, and chymotrypsinogen to chymotrypsin. These are all enzymes involved in digesting proteins. And as I said, the remainder of the enzymes do not have zymogen forms. And now I'd like to sort of put it all together and look at the contributions of both the pancreas and the gallbladder. If you look on the left, food enters the stomach, where the pH is very acid, it's about 2. And there's an enzyme, a protease there called pepsin, which has a pH optimum of 2. So it functions very well to start the digestion process. So the food, partially digested, called chyme, leaves the stomach and goes into the duodenum, the small intestine, where the pancreas is sitting right there along with the gallbladder. So here's where the digestive juices enter the small intestine, where digestion is completed and absorption of the nutrients that you get from food, the absorption process occurs. So if you go to the lower part of this, bile is produced in the gallbladder, and the pancreatic juices are produced in the pancreas, and they mix in the duodenum. Now, what's the role? We know what the pancreatic juices do, but what does bile do? Well, on the right, the gallbladder produces a complex fluid containing water, electrolytes, and organic molecules, including bile acids. And it's the bile acids that we're concerned about. Now, here's an example of a bile acid, cholic acid. It's hydrophobic. It's hydrophobic because it's nonpolar. It's a steroid. You can see that from the structure. It's a nonpolar steroid. However, if you attach an amino acid, glycine, to this nonpolar compound, it now becomes what's called amphipathic. Amphipathic means that it has a nonpolar end and a polar end. And this may sound familiar to you from what you remember about phospholipids. Polar on one end, nonpolar on the other. But this amphipathic nature of bile acids helps them, allows them to take care of fats. So bile acid has two roles. It will emulsify lipids. That means make them partially compatible with water, and then finally solubilize the emulsified components so that lipids and water can coexist, and you can derive nutrition from lipids. So the collaboration between the two is that the gallbladder secretions solubilize fats, the pancreas secretions digest the polymers, and between the two, you get complete digestion of your food. And then just to add on, don't forget that the pancreas produces hormones that regulate the absorption of food from blood to cells. So is it clear to everyone? I'll have this in your Canvas container so you can study it.
I feel great, so maybe
Dante, can you mute your mic?
Maybe I'll stay, I'll slide, so I walked up the street.
Professor, can I ask a question?
Yeah.
Is food already like starting to break down in the stomach before it gets to like the gallbladder and the pancreas?
Yeah, and that's done primarily by the acid conditions. Acid alone will start to take polymers apart. And then pepsin is a peptidase, which starts working immediately on proteins. So when the food enters, it actually, food starts being digested in your mouth, where you have, for example, amylase that starts digesting the carbohydrates. But the stomach is primarily a storage organ. And even though there's partial digestion there, it's not until it gets to the food gets to the duodenum that the serious digestion and all the polymers are broken down. And remember, an important point here is that the stomach is pH 2, that's really acid, whereas and pepsin has a pH optimum of 2, so it can work there. The other digestive enzymes have pH optima much closer to neutral, closer to 7. And so there's this huge transition in pH between the stomach and the intestine. And that's where the bile acids also play a role, because they're buffered, and the pH goes from very low to medium as soon as the food gets into the small intestine. And of course, why are we talking about this within the context of cystic fibrosis? Because these ducts the pancreatic duct and the bile duct get clogged up with mucus in a cystic fibrosis patient for the reason that I gave you in step 2 at the beginning of this, and that is chloride ions can't get out of cells, water cannot accumulate on the surface, and the effect of water is to dilute the mucus so that it can flow and be swept away. In cystic fibrosis patients, the chloride ions aren't transported. There's no, or there's much reduced water on the surface, and the mucus gets very thick and in bad cases can completely block the flow of digestive juices. So as I said earlier, you can think of it as the patient starving or on the way to starvation, because it doesn't matter how much food they eat, if it can't be digested, they don't benefit from it.
So in that way, is it kind of like celiac, where the patient is not able to absorb the nutrition even if they're eating it?
I'm not familiar with that condition, but yes, what you said would be true, that even though they're eating food, they can't digest it.
Professor,
Yeah.
this kit. What is different between secretion of the gallbladder and secretion of the stomach?
Secretion of the gallbladder versus the stomach?
Uh-huh.
Well, the gallbladder secretes bile. And if you look on this diagram here, the main component of bile that we're concerned about here is this glycocholic acid, this amphipathic molecule, which has a nonpolar end and a polar end. And that allows it to emulsify fats. So much of what we eat is fat. And in order to be able to digest that, it needs to be made soluble in water. And so the gallbladder solubilizes fats. The pancreas digests them. The pancreas has lipases to digest fats, proteases for proteins, nucleases for nucleic acids. So all the different polymers that are in the food we eat can be digested by pancreatic enzymes. But fats would not be accessible because they're insoluble in water. And what the gallbladder does is makes them soluble, makes them accessible to the digestive enzymes.
Excuse me, Professor.
Uh-huh.
Garit speaking. I have a question about cystic fibrosis. Since it's a genetic disease, can like a doctor do editing for genome so that they will fix the problem before it gets started?
Yes, people are working on that right now. Using CRISPR, it's easy to take out the mutation out of the CFTR gene. And it's easy to replace it with wild type. You can do that in a test tube, but then to get that back into cells and get it into a whole organism is much more complicated. And so that's where the field stands right now, is how to use, it's easy to use CRISPR and accomplish something in the lab. It's not so easy to translate that into a whole organism.
Okay, thank you.
Okay, anything else?
Professor, I have a quick question.
Uh-huh.
It's Marcus. It's about like the stomach and like where does like your absorption of like nutrients and like the benefits of like eating food, where does that occur?
Small intestine.
Oh, okay.
Entirely in the small intestine. So remember, if you remember diagrams of the body and the digestive tract is this very long small intestine. It's all coiled around back and forth, and then you have a colon. But between the colon and the stomach, the small intestine is a very long organ. And it is so long because that's where the digestion is completed and absorption of the nutrients occurs.
Thank you.
Okay. If nothing else, I'm going to stop sharing that.
I do have a quick question. This is Zach. We've been talking about cystic fibrosis for a bit now. I was just wondering, it doesn't seem like it causes fibroids. So I'm just wondering why it's called cystic fibrosis.
I was just reading about that a couple of days ago, and now I've forgotten. It does cause fibroids.
Just not where we're looking at it?
Yeah, I wish I could tell you more detail, but I've forgotten the detail of it. But it was, yeah, initially it was named fibrosis because of the fibroids.
Gotcha, thanks.
Now I want to talk about your project. With this document, and again, a modified version of this will be in your Canvas container soon. So here are the websites that you will find helpful in doing your project. Gives you background about especially this company Vertex, which makes drugs when it comes to talking about a treatment of your specific mutation. It's very likely that if there is a treatment, Vertex has it. Johns Hopkins has a couple of very good websites. And after you review all that background, you'll go to this website in order to do your project. And the website is self-explanatory. It'll tell you what to do and when. So for your project, a mutant from the list below will be assigned to your group. So there are 12 mutations here. You will get one of them. And you will enter your mutant in this web page and get some information that you'll need from that. But you'll have to resort to other sources as well, some of the other websites, to get all the information that you need. Your project is worth up to 50 points per student. If you did a perfect job, each student who is involved in that job gets 50 points. You will be assigned to groups of 3. Each group will be assigned a mutant, and each group will have 10 minutes to make an oral presentation illustrated with 10 or fewer PowerPoint slides. And the timing of this, because we don't have endless time in class, and this will be done during a class period, the timing will have to be precise. To make it precise, a point will be deducted for every 15 seconds over 10 minutes or 15 seconds under 10 minutes. And in order to achieve that kind of precision, your presentation will need to be reviewed. And the presentations will be done on May 26th. That's the last class period for the semester. And so I say tentatively because it could get moved to a different day, but at the very latest, it would be May 26th. Now, in order to fine tune and hone your presentation, you need to have to put it together and then get it approved prior to May 26th. And to get approval, you'll need to make an appointment with Scott. And 10 points will be deducted from the score of any presentation that is not reviewed by Scott. So Scott, would you like to say something about the reviewing process?
So the whole idea of reviewing is to make sure that you are putting forth the best slideshow that you can put forth. It's to make sure that you have followed the rubric and all the guidelines. It's to make sure that you have practiced your slide. Basically, it's to give you a let me take a look at it, run through practice session before the professor sees it, and you're going to get graded on it. So we could take a look at it. You can, if we have enough time, you could present it. That should be hopefully part of the review, and then I can provide you feedback, and you can make alterations prior to the actual presentation day.
And how do they go about making appointments with you? Scott, are you there?
So by the 7th, yes, I'm having issues with my microphone. So
Okay.
by the time the April 7th date rolls around, I will also have at that point some sort of sign-up sheet for your groups. Give you 2 weeks or 3 weeks maybe to work on them before you start coming in for reviews. And they would probably be done outside of workshop hours. So that's something I have to work out with the professor ahead of time. Also, you can in workshop ask for pointers, ask for tips, things like that prior to this appointment. But basically look at the actual appointment as your dress rehearsal.
Okay, Scott mentioned April 7th. I've chosen that date because that is approximately the midpoint of the semester. And by that time, the roster should be stable so that all the students that are still on the roster at that point are likely to be on the roster at the end of the semester when your presentation will be. So if we make groups and assign mutants on April 7th, they should be valid then at the end of the semester on May 26th when the presentations will occur. So what is to be included? These are the general instructions. The effect of your mutation on the structure of the CFTR protein. The effect of your mutation on protein function. And if you remember, protein function can be affected at many different stages of the process between the gene and the membrane. And so your particular mutant will affect that process at some point. Then what are the disease symptoms caused by your mutation? And what are possible treatments for the symptoms caused by your mutation? Then the grading will be done according to how well you adhere to these instructions. The quality of the slides. Here's a website that Scott suggested that you might get good advice on how to make a good slide as opposed to a bad slide. You'll be graded on the quality of your oral presentation. And that's why it's important that you review in advance and you practice speaking in advance. And finally, you need to draw a conclusion at the end And the quality of your conclusion and how it's based on available evidence will be considered as part of your grade.
Professor,
Yes.
sorry, this is Kit. The paper you are reading is different from the one we have for projector.
Yeah, that's because I put a preprint in Canvas. It's what I had at the time. I will replace that with this.
Thank you.
Okay, anything else?
Teacher, I have a question about our last chapter about the protein synthesis and mutation. I couldn't find the chapter on the book.
I'd have to look it up. I can't remember if I assign pages. Does anybody know on protein synthesis? There's something in the book about it, but I don't remember the page numbers right now. Anybody have that?
I'm trying to pull it up right now.
For your study purposes, so for example, the quiz today is pretty much along the same lines as previously. The questions are derived directly from lecture, from the slides shown in lecture.
Does it include Monday's lecture? Just not today's, correct?
Right, it includes last Wednesday and Monday.
Yeah, correct. Thank you.